Thank you for that welcome. I feel very humble to be standing here this evening. It's a great honor to give this lecture. I'm very moved by the history of this building and this very spot. And I thought I'd start with some of the history in case you haven't been here before, because I found it fascinating when I was reading about it. And you will see I've recreated on my slide a painting that's outside. And I learnt today that one of my heroes, one of my scientific heroes, Lord Rayleigh, is sitting just here. And we talk about him every day in our lab because he discovered the Rayleigh limit for electrospray droplets. So this was a very moving thing to discover that he was at this very event. I'm also going to start with history that's very pertinent to our research. So I work in mass spectrometry, and mass spectrometry can be considered as a balance which reports molecular weights of molecules. And the first balance was, I really like this story, it was designed by Antoine Lavoisier, and it's shown on the right. And he used it to monitor chemical reactions. So if you changed the molecular weight of a molecule, he could weigh these reactions very accurately and tell you what had happened. So this was a very important step in chemistry because it was one of the first ways of monitoring chemical reactions. He, as you may know this story, his wife at the back in the beautiful dress and the blonde wig, she always illustrated his scientific papers and translated them from French to English. And he was later guillotined, sad story this, for falsifying the weights of tobacco. But she had quite a good life because she then went on to marry Count Rumford, who, of course, was one of the founding members of the Royal Institution. So there's a sort of cyclic thing to this story because she was pretty much misunderstood, stood in the background always, but it was really the power behind a lot of very important scientists because Count Rumford discovered the third law of thermodynamics, and her first husband, the first chemical mass of reactions. So she was actually more important than people gave her credit for. And it sort of makes me think that women's roles in science were often misunderstood, and hopefully that no longer happens. I'm also moved to be standing here because when I was a student, I had to do a project on J.J. Thomson for my PhD because he was arguably the discoverer of the first mass spectrometer. And he stood here, I find this quite incredible, and unveiled the discovery of the first particle beam experiment whereby he deflected electrons. And he said that in one of his Friday night discourses, where he stood here in a black bow tie, he said, I've discovered this small particle, which I'm going to call a corpuscle, and I can see that it deflects in magnetic fields. And so there's a huge amount of history from this very spot. And while I was a student at Cambridge, I went to see the very first mass spectrometer that was assembled by J.J. Thomson and his student Aston. And this, of course, is still there. And if you look very carefully, you can see that there's a place where gases were admitted, and then it had to be sealed. And then there would be electrons, an electron beam, and there were magnetic deflectors here. And then this is one of the very first mass spectrographs. And what he did was to separate the isotopes of neon, which is just atomic mass of 22, and these are shown here, separated by a magnetic field. So this was really the very, very first mass spectrometer. An atomic mass of just 20. So I'm going to bring this now a bit more personal and tell you when I started in mass spectrometer, it looks a little more modern than that, but not a lot. So this is when I sit here on the left in a bad fashion state, perhaps. But this was when I was a technician at Pfizer. And I actually like this picture because there are 4 men on the side there actually fixing up the mass spectrometer, but I was the one who got to drive it. So I felt very proud of this. And to this day, I still have a huge fascination of seeing a beam of ions fly through a mass spectrometer and what you can learn from that experiment. So I thought I'd introduce you to the sort of levels of molecules that we could study at that time. And I chose one that you will all know, which of course is aspirin. And I'll show you what the mass spectra used to look like. So this is aspirin. It has a molecular weight of 180 daltons. So that means you sum up all the atoms and you have a total mass of 180. And we measure this on this chart. And you'll see that actually the peak for 180 is quite small because in those days, the way we put molecules in, they fragmented, they broke apart to give all their components. So you can see that this peak's quite large, this peak's quite large. But what this did was to give us a sort of fingerprint and an interpretation of the groups here. So we could start to say, oh, it's cleaving across here, it's breaking these bonds. And for me, I really loved to do this because it was a bit like a Sudoku puzzle where you start to get all possibilities and then you can solve the problem and you feel tremendously satisfied. The molecules became a bit more challenging than aspirin, but that's the sort of thing we started with. So I spent 7 years of my life at Pfizer doing very small molecule drug experiments. And then I was very fortunate to go to Dudley Williams, my supervisor, who very sadly died recently, and he introduced me to much bigger molecules, and we started to look at the interactions of vancomycin with the bacterial cell wall. I'm not going to go into that in any detail, but just to say that my role at Cambridge, I was doing a different project. I was looking at how we could sequence peptides, which are parts of proteins. So these are the amino acid backbone of proteins. And the one I was looking at was purified from a fish. So you will know, if you have fish at home, that as fish swim through different coloured backgrounds, they can change and adapt to their surroundings. And we believed that this was known as the melanocyte stimulating hormone. And at the time, there was not a sequence. And so this is actually taken from my PhD. This is in the old days, you had to type these things up. And this is a spectrum of the melanocyte stimulating hormone from a fish. We purified it from a fish, and I was quite surprised that we would get such a good spectrum. But the masses between the peaks, as I showed you in the aspirin, they're very diagnostic. So for the aspirin, they told you about the side chains. For this peptide hormone, they tell you about the sequence of amino acids. You'll know that there's 21 different amino acids naturally occurring, and this gives us a code from which we can read a sequence. So we can see that it's serine, tyrosine, serine, etc., etc. So we can, by looking at the mass spectrometer, we can see. And this was actually quite ahead of its time. This was just the very start of peptide sequencing. Nowadays, it's a huge field of proteomics where people can read off whole proteomes and from cells. They can read all the proteins that are in there. But this was when I started at Cambridge. This is proof I was there. And actually, I then did something quite unusual, which was to take quite a long career break. And I didn't really follow up, as you don't when you have children at home. I didn't really follow up what's happening in the field. And when I came back, of course, there was the internet, so I could Google what happened. And I thought, I wonder if anyone ever cared about my sequencing of this hormone. And all the hits that I got were to, unfortunately, Michael Jackson, because allegedly he also used this hormone. I'm never sure if this is true or not, but to affect his own whitening. So it had a history that was actually quite important. So while I was away for my career break, which was of 8 years, some revolutionary things happened in mass spectrometry. So we didn't just go from looking at peptides and sequencing the amino acids along small chains. It went to whole proteins. Now, whole proteins are huge in terms of mass spectrometry. So they're 20 kilodaltons, 30 kilodaltons. So instead of being just 180 like the aspirin, they're 20,000, 30,000. So these are very big molecules. And they were awarded a Nobel Prize for chemistry for these mass spectrometry techniques that could get these really large molecules into the atmosphere of a mass spectrometer. So one of my heroes, John Fenn, he received part of the prize for the discovery of Electrospray mass spectrometry, which I'll explain to you in a moment. A quarter of the prize for moldy mass spectrometry, which you may have heard of, but these are very important techniques nowadays in the whole proteomics era, and also for NMR. Don't talk about that so much. But actually, I rather liked what John Fenn said, because, of course, the Nobel Prize attracts a lot of attention. And to describe his work to the press, he said, well, I gave molecular elephants wings. So he got these very, very large molecules that you wouldn't ever imagine flying. He got them to fly through the gas phase. So this is where the change of phase comes from. You put things in and they're in solution, but then they fly through a mass spectrometer, and that's the magic of the whole technique. So how does it actually work? Well, what we do is, and we still do this, we take a glass tube, which we fashion in our lab. We have a very fine spray of droplets. And then when they get to the Rayleigh limit, which is why I like Lord Rayleigh, they actually emit protein molecules. So if you look here, this is getting down to the Rayleigh limit when protein molecules are emitted, and then they fly through the mass spectrometer and we can detect them. So this is a rather crude analogy, which I hope you'll forgive, but it's really rather like creating a sneeze. So you have a spray of tiny droplets, and within those, as you know in a sneeze, you have virus particles. Well, it's very similar. You have protein particles or proteins within these droplets, but rather than passing them around, they go through the mass spectrometer and are analyzed, and spectra are recorded. I think that's not a very tasteful analogy, and this is a much better one. This is from the Escher painting, and I believe this really depicts electrospray. It's taking something that's happy in solution and not changing it too much, but actually giving it wings to fly through the atmosphere of the mass spectrometer. But it's still staying intact, and it still has some resemblance of what it looked like before. So the first problem that I was keen to tackle when I came back from my career break was actually the protein folding problem. So how does a linear chain like this, how does it fold? So if you know about proteins, they fold into helices like this. This is actually a toy for attention deficit children, which I find quite interesting because I have it on my desk and find it quite therapeutic. I don't know what that says. But the protein folding problem is actually an interesting one because proteins fold to a three-dimensional shape, but it's not clear, or it wasn't clear, if they all fold in the same way. So some of them might fold from one helix and then one beta sheet region, which is more pleated. Or it may be that both ends fold together and they somehow collapse in the middle. Or this bit could remain unfolded until this bit folded first. So that was a question at the time that we didn't know the answer to. So we thought we could use mass spectrometry to really try to get to grips with this. And this, my mentor, Chris Dobson, Sheena Radford, and Andrew Moranka, when I was in Oxford the first time, we were looking at the protein folding problem of hen lysozyme. Now, if you have an egg in the morning, you'll have egg white, which contains this protein, hen egg white lysozyme. And this is what we were studying. And we looked at how this unfolded structure formed either helices or helices and beta sheets. These are the pleated regions. And we could see that we could trap these. And the way we did this was by using a hydrogen exchange labeling. It doesn't matter if you don't quite get that. That's a change of mass. So if you have, for example, if I have my protein unfolded and then I sort of spray paint it, it would take a lot of paint. If I folded it up in a very compact structure and then I painted it, I wouldn't get so much paint. And that's exactly what we do. We spray it with deuterium, which weighs more than protons. And then if it's exposed, it'll exchange or become covered. If it's tightly folded, it won't. And so we can read that off by changes in mass. And we fold the thing completely, and then we can see if it's heavily deuterated, partially deuterated, or not at all at the various folding times. So in this experiment, we had a heavy protein. Once it had folded, we were unable to paint away any of that structure, so it retained that heavier mass. And in the middle, we've got half and half, and at the end, we've got not folded at the time of the spray. So this was actually quite exciting at the time, because this was the first folding experiments that had been done by mass spectrometry. But that, if you do something for the first time, there's a lot of people who don't believe it, sadly. So there was criticism of that experiment, because people said, well, how can you measure folding in a mass spectrometer? It's a gas phase. It's a change of phase. You've gone from solution, where proteins are happy, into the gas phase. That can't be meaningful. And people kept saying, well, protein folding takes place in the cell, which is this really crowded environment. And it's very likely that molecular chaperones will be involved. And these molecular chaperones are, where are they? These sort of tubular-like things where protein initially comes off the ribosome and then has to fold with inside. And it's kind of, I can't quite make one that I can hold up for you, but if you have several proteins around, and then your folding protein is threading through the center, and somehow it's providing a protective environment for that protein to fold. So these are known as molecular chaperones that actually promote protein folding. And sort of the chaperone idea comes from the protection that you'll have heard of with chaperones. It's a sort of very old-fashioned idea now, isn't it, to have a chaperone? But chaperone folding means I'll take care of you while you're folding in the center here. So it's a sort of protective environment. So we started to look at how this protected environment would change folding. And we used a molecular chaperone, and this is supposed to represent these blobs here. It had, at the time, well, it still has 14 subunits, so 14 of the same protein that form these two rings, and inside would be this folding protein. And again, we use this labeling in solution, obtained a mass spectrum, and measured the mass of the piece in the middle. And then using the same experiment, we could say, is it folded or is it not folded? And we thought this was tremendously exciting. Everybody gets excited by their own experiments. And we were proposing all sorts of things. This new phase for structural biology where you could take things into the gas phase and explore them in ways no one had looked at them before. Because on the left, a solid structure, and if you've looked around here, you'll see it's incredibly famous for crystal structures, which of course are incredibly informative. And solution structures come from NMR. This is an NMR, and this is also a very powerful technique, very expensive one as well, but very powerful. Mass spectrometry is a gas phase technique, which was considered somewhat crazy to be looking at these proteins in the gas phase. And around the time of my first experiments, this was published, which says, biomolecular folding in vacuum, crazy idea. and then if you read down, at the molecular level, life as we know it goes on in water. So it was really quite a damning commentary, and it was in a very respected journal, the Proceedings of the National Academy of Science in the US. So actually, it was very hard to publish because I would always get this cited when I submitted my research papers. People would say, well, haven't you read this? It's a crazy idea. I'd say, yes, I know, but I really believe it. And I started to think, how could we really prove that these large assemblies that I'm forming here, that they really could stay together in the gas phase? And this encouraged us to start our first high-mass quadrupole time of flight. And I'm sure most of you will not be too interested in the details of this, but the idea was that we would change this region here. Typically, it takes particles up to about 4,000 mass to charge. We thought we'd be a bit revolutionary and we'd go up to 32,000. That's a huge jump. And I remember people sort of cautioning me against that, saying, just go to 8. And I said, oh no, 32 would allow us to do so much. And I got one made, and I bought it. It's actually quite heavy. So this is the quadrupole inside that mass spectrometer that I'm showing you, that just that first region there. But this is a high mass quadrupole. And the way this is different from a normal one is the RF voltage that's applied is very much higher. It's quite a heavy piece of kit, actually. I broke one of my bags bringing it. But anyway, I bought it to show you just the scale of how ions go through here. There's 4 poles. You probably can't see, but you can come up and have a look at the end if you're interested. And we put paired voltages across these, and then the ions sort of go through in a cyclic motion, and they're also pulled forward. When we did this, the very large assembly that we had here, that was our folding assembly, stayed together. Now for you, you will probably think this is a very boring mass spectrometer, but I remember to the day when I got this, because it was such an exciting moment, because it meant that these things that had always been predicted to fall apart, being too hydrophobic, not stable enough to survive in a different phase, they actually did stay together. And we could record a mass. So this is ugly, it has to be said, and my students will not be impressed because this is not how it looks today. But most of the mass spectra had been recorded at this end before, so below 1,000, and this is now really very large for a mass spectrometer. And I remember quoting this mass, and the reviewer said, well, that's ridiculous. You can't measure something like that with that accuracy, which of course is true. But it now looks like this. And I think you'll agree that's a huge improvement from that to that. That's just the same structure. This is still the 14mer that I was trying to assemble here. This is still this 800,000 molecular weight species. And you can see it's really nicely resolved. This has got 67 protons all over the surface. And this next peak will have 66, 65. And because we can measure this now much more accurately, we can start to look at molecular weights. And that was a very exciting moment. Of course, probably just for me and a few of my friends, not so many people found it exciting, because there was by that time a crystal structure, which of course looks amazing, and you can see where all the atoms are in 3D, whereas I'm just telling you that they're staying together and they're flying together. Why is that interesting? Well, for me, the reason it was interesting and exciting is because it meant that we could take things that didn't crystallize, and we could start to see the shapes and the building blocks that were within those. So the shapes became very important. And you can think about this as proteins can have different conformations or different shapes. I like this kind of weird vegetables that you can have become very trendy. Apparently you can get these in restaurants. If you go to the right restaurants, you can get misshapen carrots and things like this. But of course they're the same material, they just exhibit different forms. And we wanted to see if we could take proteins that had different weird shapes and separate them somehow. And I'll show you how we do this, as this is a nice movie. So we put in here our protein in solution. It comes through our high mass quadrupole here, which is the pit here. And then it goes through the second part that I'm going to show you in a moment in a very transverse sort of way, and then down through in a sort of reflectron that measures their mass. And this, which I also brought with me, one I had earlier, this is a T-wave separator. So what this does is it takes the ions on a sort of traveling wave undulation, and then against it, it sends a current of gas. So if you imagine a cloud chamber or something similar, you're moving forward. Now, if you're very tightly folded, you can go through more quickly, but if you stick your arms out, it's a bit like running a race. You don't want to be unstreamlined. You want to be very streamlined to get through here quickly. So each of these goes every 5 plates, has a voltage, and then we move the voltage along, so we effectively propel the ions through. This has got quite a bad ion burn. Too many samples have been through here. Time for a new one. But that effectively allows us to separate things according to their shape. And the first time we did this, we used a really quite inspired, it turned out, example whereby we had something that looked like a polo mint, so a nice round ring of protein subunits. There were 11 of the same proteins in a ring, and then we deliberately made it fold in on itself. So those two things, one's a ball and one's a polo, if you like, and they separate differently. And for me, this was part proof to the people who said that, this couldn't tell you anything about folding, because we now could distinguish something that kept its folded shape from something that had collapsed. So this was quite an important thing. I like the carrots. So the long carrot and the partly folded carrot go through differently. So we felt that we'd proven that protein assemblies could stay intact, very large assemblies could fly, and that we could also separate them according to shape. So there must be something interesting we can do with this rather than just trying to show that we can be clever with a mass spec. We thought maybe we could answer some really key questions. For example, how do proteins come together in complexes? How about proteins that exist in cell membranes? These are hugely important drug targets, so it would be very useful if we could get into those. And can we adapt our approaches for drug discovery? And I quite like this, because this is what I'm like when I get a piece of furniture to assemble, that, if you want to find out how proteins assemble, it's actually not that easy. If you're trying to read the instructions and you try to make something, it's actually quite hard. But what we have found is that we can look at quite a few different assemblies, and I'm going to start with a very simple one. And the analogy here is that you can have one protein, but it can be in many different sized complexes. And it turns out to be quite important, because for the eye, the alpha crystallins in the eye distinguish between a healthy and an unhealthy eye. And you can see this in the picture I show here. This is clouded by cataract and this is clear. And we can now see how these proteins are assembled. And what you see here, this is rather like the grapes. So these are the grape bunches with 20 proteins, all the way up to 40 or 50 even. But you'll see that the distribution is pear-like. So this sort of dagger-like thing means that actually a healthy lens should have even numbers of the grapes in the bunches. So although the bunches are different sizes, they actually interact with each other. So this is a healthy lens. We also wanted to see if we could predict how proteins assemble into complexes. And to do this, we actually deliberately destroyed them. So we had our assemblies, and what we did was to think, okay, if we start to disrupt them, maybe they would fall apart in pairs, or they would fall apart in threes, or any other combination. And we could tell from these building blocks how they were assembled. And this we did back in 2013. We showed from the mass spectrometer all the different building blocks that would eventually go on to form much larger and more complicated assemblies. So we then showed that there were only 3 main methods to form the huge myriad of protein complexes that exist. And they come about either through dimerization of proteins, so 2 form together, this is dimerization, 2 forming a dimer, through cyclization, so you could get a trimer or a ring of proteins, and then just through straightforward complex addition, so you could get protein subunits adding to each other to make more complicated structures. But actually, if you just consider those 3 different steps, you can actually make almost anything, and you can start to predict proteins that we haven't yet found. So this was quite an amazing breakthrough for me. But what about proteins that exist in membranes? And for years, I'm going to now stop and take a quick chance at my demo, which may or may not work. So proteins that exist in membranes exist in very sort of greasy hydrophobic surfaces. So they're very different to the soluble proteins such as your egg white, the proteins that you're familiar with. And it's because they're in different phases. Remember, the title of my talk was about phase transitions. I'm usually talking about going from solution to the gas phase, but here I'm talking about an oily phase and an aqueous phase. And if I'm lucky, I can break these tablets and put them in, and it should work. And you should see this beautiful mixing occur. Otherwise, I'll just take the tablets and forget about it. But hopefully it will work. Let's see. You should see something that will remind you of your youth when you sat in your room as a student and watched your lava lamp while you played loud music. Or maybe you didn't. Maybe that's a generational thing. It's not quite as good as in my kitchen because I had much nicer bubbles. These are streaming bubbles, whereas I wanted some nice big bubbles. But the reason it works, of course, is because gas is given off and is trapped in the water, which then rises, and you'll see this unmixing. It's really not doing as well as it did. So maybe I'll just take the rest of the Alka-Seltzer and be done with it. But you can see it's still not mixing, but you can see bubbles going up through the oil. They're not as big as I would have liked, but I was going to then destroy it, because what happens in our experiments is we struggled for a long time to do mass spectra of things that were in different solution phases, although we can get them into the gas phase. But a membrane protein presented a lot of problems because it's not very soluble. And the trick to getting them to work, of course, is to use a detergent. But seeing as this is not quite as I expected, if I add detergent, you'll start to see the mixing of these 2 layers, as you know. And that's because we've broken down this surface tension, and we can start to solubilize the oil and the water. And this, it may just all bubble all over the top and make a right mess, but we'll see. I was not allowed to do anything dangerous, but I was allowed to do something that's kind of kitchen-like. Here we go. Or maybe it will just look pretty. But it should now get rid of these 2 phases. Perhaps we need a bit more. And right, it's starting to go down a bit. I'll leave it while I carry on talking and try not to spill it. Hopefully it won't explode. I don't think so. I didn't have to fill out a risk assessment, so I'm hoping it's going to be okay. And but actually I wanted to look like this, because this is really what happens when you have membranes and they're surrounded by these are lipid rafts, and this is a view of what's happening in a cell. So I found this a very fascinating thing. This doesn't look quite the same, but you can use your imagination. If I go back to the commentary that haunted me for most of my life, that it did predict, actually, that although taking things from water into the gas phase would be a crazy idea, taking things from membrane into the gas phase would be better, because the membrane is a very hydrophilic environment. hydrophobic environment, rather like the vacuum in a mass spectrometer. And I owe this really to Nelson Barrera, who came to the lab. And I quite like this story, and I'm sure he won't mind me saying, but he came not knowing anything about a mass spectrometer, which is quite enlightening and liberating, I think, because he said, well, I've worked a lot with membrane proteins. I don't know anything about mass spectrometry, but I do know how to treat them. And he got them into the gas phase using a really strange set of conditions for us. But when I saw what he was doing, I forgave him, because I thought, oh, that's amazing. You've got the beautiful spectra. Well, beautiful to me, it's in the eye of the beholder. But this is our first mass spectrum of a membrane protein complex. So why is that interesting? Well, because, as I mentioned, they're very important drug targets. They're the key, and they guard all of our cells, so they're really critical complexes, and they're incredibly hard to study because of this whole phase issue where they sit in the oily phase and they don't want to talk to the water phase. And what we did was to find that if we coated them in a detergent, so if we coated them in bubbles effectively, we could put them in. Actually, I like to think of this as going in a giant soap bubble, this Electrospray bubble, and then you release it from there, and miraculously it stays intact in a folded state. So it's effectively like this. We take our membrane protein into the gas phase, and we release the detergent. This red shield here is really protecting it as it goes into the gas phase. So what can we do? What could be really exciting? And when I was a student in Cambridge, I was always fascinated by these molecules. So as you're sitting in the audience, these molecules are synthesizing all of your ATP, which is your energy currency. And I find it quite amazing that they're sitting there rotating away 500 times a second. It's an amazingly efficient machine. We can't make things quite as efficient as this. But this part here is in the membrane, and this part here, so this part would be in the oil, and this part would be in the water. But we put detergent around it so that we can protect those phases and get them into the mass spectrometer. And the thing I like about this, because people say, well, why bother? Why don't you just do EM or something? But if you think about in a mass spectrometer, it's not in solution and it's not in a solid as a crystal would be, so it's not constrained. It has this amazing freedom of movement, which I rather like. So I was telling this analogy on the radio. If you try to run through a swimming pool, it's really hard work, unless you're super fit, but these people seem to manage. This guy, I'm not sure how he does this, but he's in a bubble, and I think it's almost into the gas phase. But if you think about running, you don't want to be in a swimming pool. You really want, if you want to express yourself, you want to be out in the air. So I think if you have your protein molecules in the gas phase, rather than seeing it as a disadvantage, that it's not the same as in your cell, you should see it as an advantage because they have a freedom of movement, they can express themselves, and you can learn something from that movement. And so this is actually my very first spectrum of the ATP synthase, which I'm going to show you in its glory, and also in its hard copy, because I think it's kind of nice, for me at least. So it was one of those spectra which, if you'd started as a technician at 16, you would never have dreamed you'd see. So, and I actually printed it because I loved it, and in Technicolor. And this is actually the original, which I found in my filing cabinet because I got my coloured pens out and assigned it manually because I could and because I wanted to. And I remember it took about 3 months because there were too many ambiguities and I couldn't really work out exactly how many subunits were in the various parts, and then it was also complicated by small molecules that were binding to the part of the membrane and also to the top. But eventually we solved it, and this is me and you who got this beautiful spectrum, and for a long time this was my favourite all-time spectrum because this rotary motor has 29 subunits, some in the membrane and some in the cytoplasm. It rotates, it has unknown stoichiometries of small molecule binding. It was a huge challenge, and is almost a sort of landmark for me, because I would never have predicted something like that could go into the atmosphere of a mass spectrometer and stay together. But it did. And these are the peaks of the intact at about 12,000. And what was very nice was that this part, the membrane part, just came away as one intact species. As I say, months of my handiwork, it was hard to understand what was happening, but I'm very grateful to Nina for working out a software assignment package. It's quite a laborious one, but it allowed us to work out exactly how the spectrum was formed, so we could take a summation of all of these simulated spectra and get the pattern at the top. Why is that even interesting or important, you may be thinking? Well, because this particular motor, as I say, we didn't know how many subunits were there. We certainly didn't know how many lipids were there. Now, the lipids are the sort of greasy molecules that reside in the oil phase and sort of help grease these molecules while they're sitting there. And we found that in this particular spectrum, there were 5 lipids and 1 nucleotide. And this was quite informative, because we could then propose different ring shapes. And we found that the lipids fit rather like the shape sorter. So the red is the membrane part, the green is the lipid, and the blue is the part that it turns. So the membrane subunits would all sit in the membrane, but between them, interdigitated here, would be the lipids, which actually sort of grease the wheel. So if you imagine, if you have a motor, a rotating motor, you're going to have to lubricate it, and the lipid molecules are what lubricate it and help it to turn the rotating shaft. And the mass spectrometry was very important because it told us exactly the number of lipids that were there for proteins. We went on, this is getting a bit more technical, but we went on to look at how these now move, and we used ion mobility. You remember the separation through here, to show that these molecules actually have quite a flamboyant movement. As I said, freedom of movement. They can kick their arms out quite happily and wave. And these are actually doing this in the mass spectrometer. And if we then stitch it together with the base, we get this sort of wobbly drunk motion that is characteristic of the rotating ATPase. And we know this because this particular subunit is capable of quite a lot of different movement. And if you stitch the whole thing together, you get this sum of motions which lead to the rotating motor with a wobbly head. When you've had a few too many drinks, that's what people look like. Actually, one of my best experiences, I think, when I was lecturing, I presented this talk and someone went away and made a movie. So I was very impressed that he listened so intently. And this is the rotary ATPase, and this is its regulatory motion, because when circumstances are not good, it dissociates, it loses its head, as you see there, and the lipids come in and form this plug around it so that it stops the gradient. I'll just show you that again because I talked too much over it. But as it rotates in the membrane, this is the membrane at the bottom, and you'll see that it breaks. And we could mimic this in solution by adding adverse conditions and monitoring the dissociation of this complex. This is it passing protons across here, and then you'll see that as it breaks, it actually stops, and it has a sort of mechanism that we're able to study. Okay, enough of the rotary ATPases, let's get to some drug finding. And to do this, we need to find ways of monitoring the stability of membrane proteins. And we wanted to use Electrospray. We want to bind it to small molecules. We want to then desolvate and then energize these molecules and see how they unfold. Now, if you imagine that you're tightly folded up and you're bound to a molecule somehow, then you would be much more stable than if you had no molecule bound to you, in which case you could unfold more readily. So we want to measure unfolding as a function of what's bound to the protein. And I'll show you what this looks like, if this works. As we start to unfold, you'll see that these molecules start to stretch up and increase their collision cross-section as they go through this region of the mass spectrometer. So then, when they're unfolded, they're not running in a streamlined way, they're going more slowly. So we can measure that and it will tell us how stable the protein is. And we did this for one particular protein, known as the ammonia channel. And you can see here it unfolding in response to small molecule binding. I won't go into the details of this. We could see which small molecules gave it the most stability, and then very fortunately we can then knock out that binding site and see that the stability goes away. And this led to our first, my first, not their first, crystal structure, which I've put here because I'm very proud of it, it's my first one, of the ammonia channel in its correct lipids. And why is that interesting or important? Because if you see in this region, this is the lipidic structure, the one bound to the right small molecule, whereas the other one is the one where detergent is added and it turns its arms in. So if you have the right lipids, it's rather like floating on the membrane. So you stick your arms out, which is in fact these large, bulky tryptophan groups here. They turn out when you have the right membrane environment or the right lipid. They turn in when you have detergent because they don't really like the detergent. So this, for me, was mass spectrometry informing crystallography, which was a big turnaround because we'd always had crystallography saying, oh, that's the bee's knees, and mass spectrometry was a correlation. But for this time, we were the ones influencing the mass spectrum, the crystal. So this, for me, is one of my favourite moments when in science, and you have to cling on to these when things are not working, because these are the great times when we were able to have the front cover to show this molecule really flying through the mass spectrometer and being then crystallized with the right lipids attached. And you can see here, they keep falling off because they're magnetic, but the lipids actually, in real life, of course, are not magnetic, but stick on at the sides here and actually influence the structure. So this was exciting, but of course people didn't believe it, as usual, so it's a new thing. And so they said, oh, you've got to do all the solution experiments that prove this is the case. And we tried endlessly at the top there. You'll see we did all these different experiments, thermally unfolding the protein to see which molecules would stabilize. And none of them looked any different, which is why, with the mass spectrometry, the bottom, this is incredibly powerful, because if you unfold just a bit in the mass spectrometer, that is read as quite a big change. So if you're going through the mass spec and you just stick your arm out, that makes a big difference. Whereas if you're trying to measure that and you're heating up, you don't really know what's happening, and it's very much harder to see these subtle differences. So now we've got in place, we can fly these molecules, we can see changes when small molecules bind to them. We should be able to do something with drug discovery. It's a theory. So one of the major problems, of course, is drug resistance and antibiotic resistance in particular. And the way this works, of course, there's many different mechanisms, but one of them is through efflux pumps, which means that they kick out the very drugs that you want to put into the cells that you're trying to treat. So if we now go and look at this particular ABC transporter, it's an efflux pump, it's known as PGP. This might work, I hope it will. So if you look at this, this is the membrane, this is the greasy part of the two layers. The molecule comes in through the side, through the membrane. There's ATP binding and it kicks out the molecule. So if you think, if you're trying to treat a cell and this is happening, this is very unhelpful. It's effluxing the very drugs that you want to keep in the cell. But for us, this is a difficult experiment, and it didn't work for a while. And why is that difficult? Because we have a huge excess of detergent and we're looking for one small drug molecule. I liken it to this picture, where everyone looks the same except him, and we're just looking for him, basically. So we have 100-fold excess of detergent molecules, and then we're looking for this one small molecule that's binding. And we can find this yellow and blue, bound to our protein molecule. But really we want to see if we can see it stopping the fluctuations. The fluctuations we can measure with our eye mobility separator. And we can see how different molecules can add to the opening and closing. It's rather like a Venus flytrap. So if molecules are coming in, are they forcing it open or is it remaining closed? And this is what we're doing here. We're measuring the mass spectrum on the left and the drift time, which is through here, to tell us about the conformation. So if it's in these two different conformations, this one that spits all of the things out of the cell, which is not helpful, is smaller than this one. So we can separate those two. So we can see here that we have predominantly, we have this one, and then as we go around, we moved this outward effluxing pump. So it's unfortunately pushing out all of the drugs that we want to keep. And we can see as we change the molecules that we're adding, how we go around this cycle of moving it into the more open outward form, which means that it's moving out all of your therapeutics that we're trying to keep in. So that's one mechanism of drug resistance. Another one is to think about how lipids might hold open the outside of the wall here and allow antibiotics in or out. And we found recently that if we look at how membrane proteins are affected by charge, we can see differences in opening and closing. I'm not going to go into this in too much detail because it's quite complicated, but just to say that we can see lipid binding under different phases. And you can see here, there's a big change in this protein on PEF. So it binds PG very readily when we've got protons around, but not when we haven't. And we conclude from this, and we confirm this with electrophysiology measurements, that actually the PG lipid is stabilizing it in an open form. Why is that interesting or important? Because that's the form which drugs can go through and into the bacterial cell. And a way to test this theory is to see if we can thread an antibiotic peptide through this particular ONPEF and see if we can see that this goes in more easily if we have the right lipids surrounding it. And this is the result of this experiment. We can then start to study drug binding to ONPEF and vary the different lipid environments. And we can see that in the case of POPG, we can keep these channels open more readily than in the case of POPC. So this, I think, is very exciting because it tells you how lipids influence drugs coming in and out of bacterial cells. One of the last stories I'm going to tell you about is one of the things that is actually quite difficult for drug companies to study, I think, and this is one area where I think we can really add some new information, and that's the side effects of drug binding. You'll know that lots of drugs target membrane proteins. Membrane proteins are these very greasy things that actually don't have much specificity. They're very hydrophobic, and it's quite hard to target them selectively. And the story I'm going to tell you about is about anti-HIV drugs that bind to this metalloprotease, and they're thought to influence lamin diseases. This is proper lamin, this is improper lamin, and unfortunately, if you have this, it causes Huntington's progeria, and this is a disease of improper trafficking of lamin, and it's thought that anti-HIV drugs bind within this cavity and then stop this proper processing that should go on of lamin peptides. It's not been shown by molecular means previously, so for me this was an exciting result. in that we could see that these two anti-HIV drugs, ritonavir and lopinavir, actually bind quite tidily to this protein, and the ones that are thought not to bind, these two at the bottom, actually don't. These other funny blobs here represent lipids and cholesterol binding to the protein, but not the drugs. So the drugs that are implicated in progeria actually show up in our mass spectra as binding within these cavities. And this binding then stops lamin processing, and we can see that because we can monitor a peptide, which is the C-terminal of lamin, and we can see it binding in here and turning over, so in fact catalytically cleaved, and then when we add the drugs, it no longer is active. This is the drug binding that we now map in here. And so this was really a first example for us of a drug that was targeting HIV, but was actually causing this rather bad side effect, and had been noted previously that this particular drug concoction that was being taken for HIV was causing this premature aging disease. And just finish with some very recent results on our new exciting high mass, high resolution mass spectrometer, because to date what we've tended to do is to add lipids and then question what they might be telling us about protein stability, protein structure. But that's sort of cheating in a way, because what you really want to do is see how natural lipids might be moving around according to the proteins that are within the membrane. So without adding anything, let's take a protein out of the membrane and see which lipids are present. And that's what we can now do. We've got a different mass spectrometer, which is so much better resolution than the first one we had, which I showed you, the first quadrupole. Works in a different way. But if we zoom in on this very unprepossessing sort of peak here and expand it, you can see that there's a huge amount of detail. And this actually gives distribution of chain lengths of lipids. So lipids are these sort of long, often long chain fatty acids with charged head groups that sit around proteins in natural membranes. But they might change this length. So if you have a very long helix, for example, these might be longer, but if they're shorter, maybe shorter. And we can now see how proteins could maybe modulate that natural environment around them to suit the pitch of their helices, because in red is the normal distribution, and in black is the distribution that we found around this particular protein, which is a sugar transporter. And you can see that this is a much longer pitch of hydrophobic tail than here. So we can now start to look at natural membranes and how they're modulated. I'm now going to sum up, you'll be pleased to know. So I started, 1913 was the separation of the isotopes of neon. My spectra of aspirin, which are not very exciting, were recorded in 72. 2008 was this, I checked the date, was this mass spectrum of the rotary ATPase. 2013 we start to get into drug discovery. So standing here, where Thomson and colleagues have stood before me, I hope that they'll think that's some progress with neon. So huge amount of thanks are due to my team, my collaborators and my funders who are pictured here. This is last year's picture. Very grateful to the Royal Society, the ERC, MRC and the Wellcome Trust, and to all my group members and collaborators over the last few years. I'm always struck by the sort of symmetry of things. I'm not sure why I am, but I am. So I kind of like the fact that J.J. Thomson stood here, and then similarly Aston, who was his student, actually from Birmingham, which I find odd. He also stood here and had the first mass spectrometer. My former supervisor, Dudley Williams, also stood here on a Friday night discourse talking about vancomycin, antibiotic resistance, and in a very strange cyclic way. I'm coming back to look at that myself, but in a different way. And of course, I'm grateful to the fact that I stand here as his student. Many of my students are here tonight, and I hope that one day one of them might be standing here giving this lecture, and I might be here still to see them. And with that, I'd like to finish, and thank you very much for your attention.